Well, good evening and welcome everyone. Let me just start by saying, Nick actually told me to give a 5-minute lightning talk. I told him that would be faster than lightning for me to summarize our results and our interests, but I'm going to try nevertheless. But let me take 30 or 15 seconds of that time to actually thank you all for being here. Thank Nick and Susan for organizing this event, and thank Jeff and Liesl for being here and for their support all this time. Nick also told me, just speak from your heart. And as scientists, we think from our minds, so slightly tricky. So just check me if I'm speaking too much from my mind and if it's not amenable to you any longer. He deprived me of my ammunition, which is my slides and my pointer. And so this just has to come from my heart and my mind in some way. But let me just also say that, this is really great to see you all here, because as scientists, when we train as scientists, when we get into science, we typically think about doing this because we want to make a difference to human disease. But then over years, we work in the lab, and every few years we make a finding and we publish it, and we hope that at some point it will touch human lives. And sometimes it happens, sometimes it doesn't. And I particularly feel honored to have had this opportunity at such an early phase of my career. And let me just say that I'm actually personally humbled by the faith that people would have that science can actually touch their lives or make a difference in such a challenging disease. And so with that, I will switch to my mind, and I will tell you more about what my lab does. So my lab is interested in immune responses and in immunology in general. We're interested in how the immune system functions in many different scenarios. So I presume you all know that our bodies are built to fight infections and autoimmune diseases, but that's not always the case. The immune system is like a double-edged sword. You need enough of it to be able to fight the infection or to get what is called host defense. But if you get too much of it or you get too little of it, you can cause something which is called immune dysregulation, which in some cases leads to autoimmunity. So how does this all tie into Lyme disease? Now, one of the challenges underlying Lyme disease is basically this thing that nobody understands, which I'm sure you all have experienced and understand perhaps better than me, which is that when people get bitten by the tick, they get this bullseye rash and they get what is called acute Lyme disease, and they go on antibiotics, commonly doxycycline or choose your antibiotic. But after a course of antibiotics, a certain set of individuals still continue to have symptoms, and they never completely resolve the disease, about 10 to 20% of them. And I'd perhaps love to hear about what your experiences in this regard have been. And as scientists, we classify people who have had symptoms for more than 6 months after antibiotic treatment as people who have post-treatment Lyme disease or post-treatment Lyme disease syndrome, if they actually display a constellation of symptoms as opposed to just one particular symptom. And what actually causes this switch from acute Lyme disease to post-treatment Lyme disease, in spite of having been on antibiotics, is a mystery. And that is part of what we're trying to get at in my lab. And I will also say here that there's another part of this mystery, which is people who actually don't display the characteristic bullseye rash and just go on to have more chronic type of symptoms, just things like neurological complaints, even in the absence of having the rash. And that's typically about 30% of the patients, about 70% of the patients do display the bullseye rash. So those are the people that we're studying. But this remaining 30% that proceeds to really severe symptoms and a more chronic form of Lyme disease is something that our study doesn't cover at this point in time, and a cohort that we think is really important to cover, but scientifically slightly more difficult to analyze, because in order to do these kinds of data analyses and get power in the genes or the proteins that we're associating with disease, we tend to need a well-defined cohort. But I'm sure with enough people enrolled in a study like this, we can derive that kind of power. So I told you the immune system is a double-edged sword. We're trying to figure out what happens in people who transition from acute to post-treatment disease in spite of having been on antibiotics. So how are we actually trying to do this? In order to do this, and again, stop me and ask questions if I'm becoming too scientific, we're basically collaborating with a medical doctor. He's basically a Lyme disease clinician. He's cured patients for years and years, and initially actually didn't believe in post-treatment Lyme disease, and after having worked with us, has started to believe in post-treatment Lyme disease. And I'm sure it's not only us, but the experiences of his own patients as well. But I think only goes to say how much patients and their experiences can change science, or the paradigms that we tend to stick with in science. So we're collaborating with Gary Wormser at New York Medical College, this physician. We get blood from him, from people who've been newly diagnosed with Lyme disease over the course of a year. So we collect blood at diagnosis, and we then longitudinally follow these patients. Like Lee was just mentioning in his talk, we need to get these dense dynamic data clouds at many different times post-infection to actually be able to resolve signatures that correlate with acute disease, and then with resolution of the disease, and then with post-treatment Lyme disease in patients who actually don't resolve the disease or the symptoms of the disease. And so we've collected samples out until 1 year with the intention of being able to cover this whole cohort of patients from acute disease to post-treatment disease. That would be individuals who continue to have symptoms at 1 year or at 6 months post-antibiotic treatment. We basically get blood from these individuals, as I said. The blood is fractionated into plasma. That is the liquid component of blood. Our blood also contains immune cells. These are commonly called white blood cells. They can be of many different types because they have many different roles in the immune response. Some respond very early in the immune response, and those are called macrophages or dendritic cells. Others respond later and produce antibodies, which I'm sure you all have at least heard of, an antibody response that that is the basis of vaccines in some cases. Those are called B cells, and then there are other cells that are actually killer cells that can actually see an infected cell and directly kill it. Those are called T cells, or at least a subclass of them are T cells. And there are many other cells in the immune system, so it's much more complex than I'm presenting it to you here. So what we're trying to do is threefold. We're taking the liquid component of blood or plasma, and we're measuring many different molecules that are produced by these immune cells when they get activated, that is, when they see an infection. We're taking the cells themselves, and we're seeing how they're getting activated by measuring many different proteins that they express upon getting activated. And then a third thing that we're doing is we're taking these same cells, and then we're looking at the genes that they're expressing, and how this gene expression in cells and cell subsets actually is changing over time. And what this allows us to do, or we hope it'll allow us to do, is actually get at the actual culprits or the actual genes, or let's say master regulators of multiple gene sets. This could happen at multiple levels, so I will spare you all that torture and won't go into the details. But what this will allow us to do is actually get at the genes or the gene sets that are dysregulated during acute disease that then return to normalcy in some individuals post-antibiotic treatment, and that perhaps still remain dysregulated in individuals who proceed to post-treatment Lyme disease. And so that's the threefold study that we're doing, measuring cytokines in the plasma, measuring immune cells and their activation states, and measuring the genes that are getting perturbed in these immune cells over time and how they correlate with acute disease and post-treatment disease. So this all sounds very complex, and it is all very complex, but in doing this, we haven't gone all the way and done all of this yet. Science is a slow process, and I'm sure as patients you've all learned one thing, which is perseverance. And that is one of the first things we also learn as scientists. You realize that if you're not perseverant, nothing actually happens. And I'm not the most patient person in the world, but I've learned that I need to be patient. So anyway, what have we found so far? I'll tell you a few things that we found so far, which may or may not, again, make sense to you, but feel free to ask questions. So one thing that we found is that Lyme infection is a very induces a very stealth immune response, which means that you don't actually get an immune response of the magnitude that you would tend to get during an influenza infection or during a bacterial infection, like when you get diarrhea, and we haven't enrolled patients with influenza and diarrhea ourselves, but leveraged, this is based on our knowledge of what we know are the immune responses elicited in these individuals. So we know that we get an induction of an early immune response in the acute phase after tick bite, but it's nearly not as much as what is induced by some other pathogens, both bacterial and viral. The other thing that we found is that you get this acute immune response and it largely goes away in individuals after antibiotic treatment, but in a fraction of the individuals, a subset of the immune signature persists. And that's a more complex thing that I won't be able to go into at this point, but what we're trying to understand is, does this residual immune signature in any way correlate with individuals who then go on to develop post-treatment disease after having been on antibiotics for at least 2 weeks, as our data suggests at this point? The third thing we found is that the early immune response mainly is that of a T cell subtype. So I told you T cells are a certain arm of the immune system. They can produce a certain set of cytokines. They also need to be activated for them to be able to produce these cytokines, or I know I'm using a scientific term now, so it's not cytokines, like an immune mediator that is produced by cells to attract other cells to the site of infection. And so what we know is that we get a selective T cell response, and we're trying to figure out if this could act as an early marker of Lyme disease. And the fourth thing that we found is that when we stratify the immune signatures or these immune mediator signatures by patient symptoms, we can actually reveal 3 categories of patients. One is, and how do we do that? That's because when we get samples from these patients, we also get clinical information from them. And so we have very detailed clinical information, diagnostic information on each one of these individuals. And so stratifying these patients by clinical information, we can reveal 3 different classes of patients: one who have a very mild immune response, very mild symptoms, one who have very severe symptoms to start out with, and a third category that is enriched for more chronic type of symptoms. So this category is enriched more for neuro-Lyme. And basically we have signatures for each one of these 3 categories that we're now trying to dissect out in more detail by looking at actually the cellular activation states and genes that correlate with these signatures to figure out what exactly they mean. And so where do we think this research can go? One, it can tell us whether there is an autoimmune response that is triggered during post-treatment Lyme disease, which we don't completely understand at this point in time. We tend to think that post-treatment disease might be due to persistent infection, but there's actually very, very low evidence of that, because going on patients who have post-treatment disease don't actually benefit from being on antibiotics. You can never culture the bacterium out of these patients. And so the alternate hypothesis that we're obviously testing here is there something about the early infection that just causes a certain set of patients to snowball into an autoimmune response as opposed to resolving the infection post-antibiotic treatment? And the second thing that we think can come out of this is basically something called host-based therapeutics, which is that if we could actually figure out which genes and which proteins in the host are getting dysregulated through the course of acute to post-treatment disease, we could then develop strategies to target them. And there are actually already drug target networks available out there, or drug target databases available out there. So if we could really pinpoint those targets, we might have the ability to actually interfere with them and benefit a certain subset of patients, that is, if we're able to translate it into the clinic. And so that is what we do, and we hope that at some point it'll reach people who really need it. There's a lot more to be done, of course. What I mentioned to you right now was our data based on immune cell mediators and based on phenotyping of a certain subfraction of T cells. But we still need to look at the genes, do a more intense immune phenotyping to really get at what these distinct signatures mean. But we're quite excited by our initial findings. And so with that, I will turn it over to Rob, who will tell you more about the proteomic work that he does to actually reveal signatures on the bacterium itself and how they can be targeted. And I'm happy to take questions later if anyone has any, that is.